If for those of you who don't know me, I'm a consultant in life sciences. I help companies scale and manage complexity and increase performance. Today, I'm joined by Lance Taylor, Chakra Chenolta, and Dusty Majumdar. Welcome, guys.
Hello. Great to be here.
All right, so if we could just start with you, Lance, just really quickly, could you do an introduction to yourself for the audience?
Sure. Pleasure to take part in the podcast. I'm Lance Taylor. I'm the executive chairman of SpIntellx, and I'm also the director of drug discovery at the University of Pittsburgh Drug Discovery Institute.
Very good. Welcome. Chakra, could you do a brief introduction to yourself?
Thanks, Don. Thanks for the invite. My name is Chakra Chenolta, and I'm the CEO of SpIntellx.
All right, and Dusty.
Hey, Don. Good to be back again on your podcast. Really appreciate it. I've been in the area of precision medicine now for over 20 years, been in companies like GE, Exact Sciences, IBM. My journey with AI, as you know, started when I was with IBM, and I've been fortunate to see this whole cycle over the last decade, if you will, and currently working with SpIntellx, heading up the strategy and marketing with Chakra and Lance, and really enjoying positioning AI in precision pathology.
All right, very good. Yeah, I almost feel like I should give you a frequent visitor card. I think this is podcast number 4 for you. You did one by yourself and a couple other ones, and then a couple other panels, and now this one. So thanks for coming back. And I mean, especially exciting, to get to talk to Chakra and Lance as well.
Yeah, this is going to be exciting.
All right. So maybe we just start with kind of a brief overview of this area. So what trends do you see in terms of precision medicine and AI? And Dusty, why don't I toss it to you first?
Okay, sure. I'm sure that Lance and Chakra will have a lot to say about that too, specifically around pathology. So I'll start at a little bit of a higher level. As you know, in between 2014 and 2018, there was a lot of hope, there was a lot of hype as well. And what we see now are real applications with AI emerging. And the reason for that is we are now living in an era of tremendous convergence, if you will. We have more data than ever coming at us. We have more computing power than ever before. And we have AI, machine learning, deep learning algorithms, which are getting more and more robust. So things are changing over the last 3 or 4 years. This is the time when AI is becoming real. And specifically in oncology, as you know, there's so many variables that enter into a clinician's decision for a diagnosis and treatment. And AI can play a huge role in oncology, and we'll talk more about that. For example, in analyzing complex and heterogeneous data coming from multi-omics or interomics, if you will, data integration to see a holistic view of the disease, monitor patients' response, for example. In a complex disease like cancer, you got to check many different parameters to monitor that. And there is a consensus that for precision medicine to succeed, you have to look beyond just genomics at this point of time and take into account the different omics, including the pathological observations that we'll talk about quite a bit today. In the case of solid tumor cancers, and Don, you know this, a major issue is that patients don't respond very predictably to treatment. And I think one of the estimates I've seen lately at SpIntellx is that 90% of the drugs are effective for fewer than 50% of the patients. So think about that,
Wow.
right? And immunotherapy, many think is the ultimate miracle, and that's less than 20% effective in most cases. So a fundamental driver of the lack of effectiveness of immunotherapy is the heterogeneity in cellular composition and signaling network among cells. And we're going to talk about how SpIntellx addresses that. And intertumor and the intratumor heterogeneity plays a huge role in drugs not being efficacious in many different kinds of cancer. So that's what I see right now. And honestly, I mean, it's been a great journey with AI, starting, with the initial forays into healthcare with Watson Health, and then working with a range of startups and now really kind of enjoying working with SpIntellx and getting precision pathology to the next level.
Yeah, it is for sure a very exciting time from both where we stand with the science as well as where we stand with the technology. It's just a sort of a great pivotal point. Chakra, do you have anything to add to that? I mean, what trends are you seeing in terms of precision medicine and AI as a leader of SpIntellx?
Thanks, Don. So one of the things Dusty alluded to is who do we really, what are these solutions supposed to address? And then if you think about both the clinicians and the oncologists and the pathologists who eventually use this, there's a big gap in the trust of AI algorithms and how much they trust it. And we saw a huge gap in the explainability of these algorithms. The clinicians don't like to use black box algorithms where it spits out a decision without actually taking you through the thought process for how it made a decision. And it might show heat maps, and that's a very popular approach. But heat maps have a trouble that you project your opinion on these heat maps as opposed to actually revealing the underlying biology. And so when we looked at the evolution of technology, we found these two being a big gaps of not having the explainability and how you build the trust and how do you exploit the tumor microenvironment in a more holistic sense to build the technologies. And that's where that brought us to SpIntellx. Lance?
I mean, it's really exciting. You're truly exciting, and great to know that. Right. I mean, from a scientific standpoint, and this is where I would definitely go to you, Lance, is in terms of the sort of understanding how AI is making the decision. I would imagine that that is pretty important overall, and having the ability to really understand explainable AI versus, a black box as well. But I would ask the same question, what are you seeing in terms of precision medicine in your space, and especially with AI?
So, building on the idea of explainable AI, in our discussions with potential customers going back to the beginning of the formation of SpIntellx, we were told over and over again that explainable AI would be a game changer in the field for the reason Chakra mentioned. People don't like to take direct decisions from an algorithm without understanding why the algorithm made the decision it did. So we think the future clearly is with explainable AI so that the machine learning can occur, but then the output to the end user, which would be the oncologist or pathologist or other clinician, they will be able to look at what the algorithms decided and agree with it or not, and they're still in control, and that's critical in this field.
Yeah, very important. And so, I mean, with the hype, kind of dying off around, what I would say AI, could be, and it actually becoming more of a reality. What are you seeing some of the real applications now powered by AI that are on the horizon? And Chakra, I'll come back to you maybe for the first answer on this one.
So, what we observed, particularly, I'll take the pathology field as a topic of interest here. 2017 was when FDA gave an approval for scanning a digital scanner where you can scan the slides, glass slides, and make a primary diagnosis. And this was the same time wherein machine learning, particularly in the form of deep learning, started picking up steam. And in the first generation of tools that came out from these efforts was to think of these digitized images as data and then and then these are vast, I mean, huge amounts of data that you get out of this scanning. Deep learning run the deep learning algorithms directly on these images. And then what we saw was exactly something that we already spoke about, is that there's a gap in explainability, that even though these algorithms can run on these data sets, yeah, they don't quite meet up the expectations. So with that in mind, I think where given the hype and given the reality of what people want, there are many applications now wherein you can use AI if it is just the basic counting of the cells. And that's happening in some domains, particularly in the pathology field, wherein all you need is a cell count, how many CD3 positive cells are there, for example. But that again falls very short of understanding in great depth the disease biology. And then so it was clear for us that if you want to get to the next stage, which is the precision pathology, you need these additional tools. And my take is that something that Dusty brought up, that this precision pathology will address many of these needs in designing, finding out why some patients respond to therapy and some don't. How you can build very powerful companion diagnostic tests, and so on. So there will be a range of applications wherein you imagine precision pathology making inroads.
Very good. And Lance, why don't we come to you next with regards to this? I mean, what are some of the real applications that you're seeing now that are powered by AI that are on the horizon?
So, in the broadest sense, I think there is an understanding that none of the individual omics are going to be the only part of precision medicine, but there's power in genomics and metabolomics, proteomics, and in pathology, in precision pathology. And more data will be integrated from a patient using multiple omics. And it's going to be then critical to have explainable AI to again tell the clinicians the reasons for the algorithms making a particular recommendation. And that recommendation could be a particular therapeutic strategy. It could be identifying a potential novel target for drug discovery. Could be an important and powerful companion diagnostic to a drug that a pharmaceutical company is developing. And it can also be as basic as putting together a whole patient or what kind of comorbidities exist. So it's going to play a central role.
Absolutely. And Dusty, I would ask you the same question. What are the real applications that you're starting to see on the horizon in this space as well?
Yeah, so we're going to talk a lot about precision pathology today, but if you think about the real applications that we are seeing today out there, the emergence of chatbots that actually interact with patients is on the rise. In robotic surgery, we see application of AI to reduce variation. There's a lot of data coming in, and AI can definitely help in making sure that it captures that 3D, 4D data and helps to make robotic surgeries more accurate and more precise. The emergence of virtual nursing assistants. Lance talked about the use of precision medicine across the continuum of the drug discovery and development process. So, from identifying the right targets to the right biomarkers in discovery to identifying certain populations of response in the clinical trial phase, all the way to market access to really understand the efficacy of certain drugs in certain populations and opening up patients with different clinical indications. I see application of AI throughout, this whole spectrum. And the last thing I think that Lance was referring to was this whole concept of digital twins, where now, you can take all this data that you're collecting from various patient populations or maybe even individual patients and come up with virtual patients on whom you can actually try different interventions. So I will just pass it on to you, Don, and I'll mute my.
Okay, very good. So, I mean, as a follow up, I mean, one of the things I know I saw years ago was this idea of having computer aided diagnosis in radiology, and just this the whole idea around having something help to read the images. Really, radiology and pathology were early adopters, overall of AI. Where are we going to now, Dusty?
Well, I mean, radiology, I mean, if you think about it, went into AI almost 10 years ago in quite a big way. And they have made some progress in terms of helping the radiologist get augmented with AI. One of the quotes I heard some time back was, AI is not going to replace the radiologist, but the radiologists who don't use AI would replace radiologists, or radiologists who use AI would replace radiologists who do. So, Don, I mean, that's going more and more clear with the passage of time that AI is going to be a critical component of radiology, and just like radiology and pathology, more and more digitization of images has happened over the last 20 years or so. And it's no surprise that now pathology is emerging as a key area in which AI is being deployed.
Yeah, I just wanted to bring up some of the chat that's going on currently while we're on here. We have Michael's on. He's putting on your website address so people can follow up with you there. It also looks like Manisha is on from Boston as well. So just wanted to mention them just briefly as well before we get to the next question. So welcome. And so with that, I guess, Lance or Chakra, I guess any other thoughts in terms of where we're headed next in terms of, early adoption of AI in radiology and pathology?
Well, certainly, as you pointed out, radiology has been using digitization for the last decade. And so they got into the use of machine learning and AI very early on. Pathology had a lag because it came to digitization kind of later, and it's only been in the last 4 years or so where the FDA started approving particular scanners as tools that were approved for doing diagnoses, not through the microscope, but from a screen. And that, of course, opened the door to the first generation of so called computational pathology tools. And those tools have been very valuable. And I think this next generation, which will really use more sophisticated spatial analytics to look at so called spatial biology, and then tie that to explainable AI so that the clinicians can understand what the algorithms are doing. And that gets to the point that these tools aren't designed to replace the pathologist or the oncologist, but to give them added support in terms of understanding, especially complex patients, results from a complex patient. There's a lot of pathology that's very easy to spot, something that's obviously normal or obviously cancerous. It can be done by any algorithm. On the other hand, some intermediate and difficult to identify things like in breast cancer with ADH, it takes the sophistication of the algorithms in order to pick it out. And one of the things that we're seeing happen now is that it can be used as a pre sign out tool for pathologists to make sure the subspecialists get those difficult cases. Because right now, 90% of the effort of pathologists is spent on 10% of the samples. And that's because it's more or less a random handout of the patient samples to pathologists. This can help guide the right patient to the right subspecialist.
Yeah, I mean, I look at this idea of sort of augmenting physicians who are intelligent and already applying complex, thought, to their field. And yet there's so much more information coming at everybody, with regards to all the technology that we have available, and having an assistant that can help us sort of sort through things would be important. I guess, Chakra, anything else that you would add to this point as well, in terms of, where we're headed?
Yeah. So, Don, I feel like both radiology and pathology being the key points for the patient care. With radiology, you're mostly looking for patterns that are either there or not there.
Yep.
But when it comes to pathology, you have this extra, there's so much information. First of all, the tissue is at a premium. And then there's so much more information and knowledge that you can extract, particularly indicating what might be the reason for the disease and where it might be heading next. And I think that sort of not just the diagnosis, but also the prognostic aspect of the pathology makes it very interesting, makes it very challenging for these algorithms. And I think explainability, besides being very critical, will entirely change the workflow for the pathologist in ways
Mm.
that they'll appreciate more. It's already they have to go when you have the slides digitized, they have this workflow solution that they have to log in, and then where the explainability comes in is that with the touch of a button, they can ask something. They can probe the disease, they can probe the tissue, and then ask what might be happening here. As Lance was alluding to, the places in that tissue could be atypical ductal hyperplasia, particularly if it is a breast biopsy, and there could be places where there is cancer. And it turns out atypical ductal hyperplasia happens about 15 to 20% of the population for the breast biopsy or breast cancer patients. And the trouble here is that the discordance between pathologists in diagnosing atypical ductal hyperplasia for some of the studies that have been reported recently is extremely high as 52%. So now the idea is that if it is ADH, you're definitely want to get a second and the third opinion just to confirm that it is ADH. But if that is not diagnosed, the chance of that patient getting cancer is about 50% within 20 to 25 years. So knowing this very early on and knowing that this is actually a difficult case is a key component of how these diagnostic tools will play a role in assisting the pathologists and the clinicians in the space of pathology.
Very good. And then
Hey
we
Don,
see.
one thing I want to hit upon before you go to the next question
Sure,
is,
sure.
what Lance and Chakra both alluded to. So in radiology, one of the reasons why we don't see even now widespread adoption of AI after all these years, after hundreds of companies getting into radiology and coming up with these AI algorithms, is to a great degree the lack of explainability. Now, there's a balance between explainability and accuracy, right? You can have a result that's 99% accurate, but you can't explain why because you have a black box. Now, is that completely useless? Probably not. You can still, but you need to have explainability, especially when you're trying to get to the root cause of a disease, a complex disease like cancer. And one of the critical obstacles in the widespread adoption of AI has been the perception of AI as a black box. And that's what these guys at SpIntellx are trying to get away from. So, when clinicians see the result, they're able to answer the question why. Why are they seeing what they are seeing from an AI algorithm? And I think that's critical. So I would just say that as we move along this discussion, you'll hear this theme over and over again: how explainability is making a difference.
And I guess from that standpoint, I'm going to sort of take a bit of an aside as well. So, um, why is it that explainability is important? Overall, I could kind of envision a few reasons, but would love for one of you to just maybe provide a little bit more thought in terms of what you're thinking in terms of why is explainability is so important, especially to the physician that's looking to care for their patient.
I know those Chakra and Lance can speak about pathology. I'll just say from a related field in drug discovery, when you're trying to understand complex interactions between genomic data, lab data, the clinical data of the patient, and you want to develop a virtual arm or an in silico arm of a clinical trial, you need to understand the underlying circuitry of the disease. So
Mhm.
if you don't really know a whole lot about, why the AI algorithm is saying, that this is where, you should be going, and you don't understand the underlying mechanisms, you cannot treat that disease, and you cannot develop the next generation drug without understanding the basic underlying circuitry of the complex disease. So, I mean, that's kind of one of the rationale for really understanding, going beyond just a couple of layers of the CNN network at the top and the bottom, but got to kind of get deeper into it. Chakra?
Right.
Yeah. So explainability, I think, is on its path towards causation. In the end, I think the need to understand why something happened is critical across these disciplines, across really for a pathologist. As I said, the tissue holds so much information as to where the disease started, where it is now, and where it is going next. And although they're only looking at one image or maybe several cross sections of the biopsy, that is a central theme that they're driving hard at. So, and that's where they would appreciate this additional assistance, and that's coming through explainability from the AI algorithms. And we have seen that we have spoken to with the market research that we did. It was overwhelming impression that that sort of explanations, when we showed them the demo of how explainability works, they would come back and say, this is going to be a game changer in our adoption of these systems. And the best part is that they could override that and say, you're wrong, to the machine, and let the machine continue to learn for that particular practice what kind of decisions are being made and why. And I think that sort of interaction between the clinician, in this case the pathologist, and the AI algorithms would be the key for how AI could be adopted in a practice.
So important. And Lance, anything you'd like to add to this as well?
Yeah, if we think about this, it's true for radiology as well as pathology, but pathologists in particular trained for many years to look for patterns within tissue sections. Their brains were the algorithms, and they could do pretty well. One of the challenges with looking at glass slides through a microscope is that people got very tired, and the reproducibility was not all that great between pathologists, whether they were looking at the slide early in the morning or at the end of the day. And in fact, some critical studies have shown that it's significantly not good in terms of having a dozen different pathologists look at exactly the same samples and make the same call. It can be as bad as 50%. So the ability to have an aid, if you will, and have that aid use the same language that you do. In other words, when their brains look at the slide and make a call based on their visual algorithms, then if the explainable AI machine does the same thing, tells them the answer and why, it's reproducing what the pathologist's brain was doing. This becomes then believable and they can have confidence in it. Now it becomes a true aid to the process rather than just a simple, well, we can replace the pathologist with a machine.
So important overall. Just this idea of, I think turning over some of these very sort of critical, elements of healthcare as well is from a patient perspective. I would say, I would want to know that my physician really knows kind of the background of the decision that was made at the end of the day, instead of fully believing at the end of the day whatever it was that was given to them. I mean, you've gone to school, you've done a lot to look at all of the information that's out there. Why not just double check it with a computer or with AI? So yeah, I agree. We see quite a few companies out there really trying to implement AI in pathology. Is there anything really that's groundbreaking that's come out so far? And why don't we go to you, Chakra, for this one?
So thanks, Don. As I said, I think given the nature of how the technology has evolved, starting from the FDA approval of the scanners and this immediate application of the deep learning algorithms, particularly, or the black box and other black box algorithms as well on this data, it was clear that you need this ladder to climb to the next version, which is the precision pathology. And the components of that is one, of course, explainability, and we talked about that. And the second is this unbiased spatial analytics. And here, what I meant to say is that when you run the deep learning algorithm, you can see that it misses out on some key aspects of the biology. For example, it will never be able to tell you that this is a fusion cell, wherein the fusion cell has properties of both a tumor cell and a macrophage cell. Right? And then what we realized is that having this ability for AI to recognize these additional communication patterns between cells, because this fusion cell is a consequence of one such communication. And then keeping track of how this communication changes over time tells you how the disease has progressed. So this is, and to enable that, you have technologies which do the spatial imaging of the tissue with several biomarkers now. There are platforms that can go up until 100 biomarkers. So pretty soon, this protein expression, these are mostly protein, but you can also have nucleic acids here. You'll see that the protein expression will start matching up with the transcriptomics in not too distance. And there are many studies that show you that just because you have the mRNA expression does not necessarily mean that the protein expression follows from the mRNA expression. So eventually, what the proteins do is going to be a very key component, and measuring it has been hard before, but gotten somewhat easier, and hence the technologies are rapidly progressing. So both in the AACR, we saw that with the American Association of Cancer Research meeting, and in the last 2 years, you will see about half a dozen platforms already in the market, allowing you to visualize these spatial properties. But then there are no analytics that go with it to actually fully exploit the information that you're getting. And that's where we come.
Very good. Lance, anything that you're seeing that's groundbreaking that's come out so far?
Well, I think it is important that the first generation computational pathology tools have been adopted rather rapidly during especially the last 2 or 3 years. And now there's been a great demand, and people are beginning to see the importance of analyzing the spatial relationships between cells in tissues, and in particular in solid tumors, the appearance of so-called microdomains or specialized regions that have different types of cells in close association. And that now enables us to investigate within those regions the complex biology that is driving the cancer. And I think now the challenge is how much of the pathology and biology can be extracted. And right now, we would say with the first generation tools, a lot is left on the table, if you will. And so, in a natural progression, the power of the first generation tools has yielded a great deal. Now, I think there's pressure to extract even more knowledge from these samples.
Yeah, and Lance,
Yeah.
it looks like you have a co-host over there on your side. Looked like your cat
Yes,
was about ready to join us.
and I had to notice how deftly I moved her away.
It won't be the first. It won't be the first time here that's happened. And I have Great Danes that I have to prevent from entering the room because they would completely take the shot. So very well.
And on
So in turn,
just to build on what Lance
yeah,
just said and what Chakra said, this is really a quantum leap in terms of where the first generation tools of pathology were and where Chakra and Lance have been able to take these tools with SpIntellx the last few years, because understanding the spatial network and the cell-cell interactions and discovering those unique spatial collection of cells and cell states that may share something in common, the microdomains, if you will. I think that's huge, and that is where explainable AI comes in, because now you're explaining why a particular recommendation was made instead of just saying that the black box spit it out. And we believe that clinicians, pathologists, would really trust this algorithm a lot more than what the first generation tools put out there.
Yeah, and it sounds to me like, I mean, I guess correct me if I'm wrong, but it sounds to me like spatial understanding in this space is what has been missing, or more or less where the gap has been. I guess, Lance, I'd come back to you. Where has the gap been in terms of the overall space here in precision medicine and AI?
Well, and specifically for pathology, but it's true for any of the omics, cell-cell communications and subdomains of different kinds of cell populations that are communicating are crucial in disease progression. We have some good tools for single cell genomic analyses, and you can get a lot of deep information from individual cells. The challenge there is that you can go deep on an individual cells, but how many cells are communicating, and can you get all of those cells with that kind of depth? Using pathology, which is a spatial omics approach to begin with, because you can look at multiple cells in parallel. And if you have enough biomarkers, you can define these domains. And in fact, you can, in an unbiased way, have the data tell you what the microdomains are. In the first generation tools, it's more biased. You might identify a couple of key biomarkers and then extract information from around those biomarkers. But in this second generation approach, the data itself in an unbiased way tells the user what are the domains that you should be looking for. And then it automatically identifies those domains and then can extract great depth of information from those microdomains. But once again, there's going to be a deeper integration of multiple omics. I think it will be even more focused regionally. So I think the most spatial analysis will come from the spatial biology in new precision pathology, but also being able to direct the other omic extraction of information based on this unbiased identification of key so-called microdomains, where you focus the metabolomics, proteomics, or genomic analyses.
And AI
Very
would be critical in integrating all these different complex data sets. And one thing to remember in terms of what Lance just said, Don, is that you got to go micro, real small, and you got to kind of look at the organization of cells at the macro level as well. So you got to look at the forest and the trees, not
right.
right. So that's the key here. Chakra?
So I was going to add to that sort of discussion is that Don, biology is not static, and the tissue actually tells you a story, and the emergent behavior is the key here. And you need tools that actually capture that emergent behavior, like Lance was pointing out. Some of these microdomains might be unique to this patient. And heterogeneity is not random. Heterogeneity is organized as the structural units that we are calling as microdomains. And they emerge and you don't know beforehand how to, that what to look for. And you should let the data drive for the emergence and for the capturing of these microdomains. And I think that's the key for how best to use these technologies going forward.
I mean, the best thing that I'm hearing too is that you're not only sort of looking at what's happening today, it's you're giving information for what might happen tomorrow, which really from what I've seen in my personal experience with cancer with family members is that so often you see this turn where they're treated one way and all of a sudden you see that it looks like their treatment is progressing, and so quickly it can turn around and all of a sudden can be back in a worse spot than you were originally. And so from that point standpoint alone, I definitely think that we need all the tools possible to kind of look at what might possibly happen in this world of ours.
And you need to capture that insight that could be hidden. It could be so subtle, right? So That's what spatial biology and really kind of AI can yield. And sometimes it's not obvious, but the solution may be right there. You're just not seeing it. There's a famous quote by Einstein that God is subtle but not malicious. So it's the hint may be already there.
Right. So, Lance, can you tell us a little bit about the evolution of pathology and why digitization is such a big deal?
Yes, so pathology was essentially the last imaging medical platform to be digitized. And part of that was, again, what I mentioned before, that pathologists are well trained and their brains are the computer and the AI. And they didn't believe early on that a machine could do as well as they could. But it is a fact that pathologists are human, they get tired. You look at N number of patient samples a day, you just can't be perfect on every call, coupled with the fact that there's subtleties, as Chakra said earlier, in some of these biopsies that it would be very difficult to identify. So I think two things happened. One, a new generation of pathologists came along that were used to doing video games, and so dealing with a computer monitor was simple to them. And the other thing was the fact that in some well published studies, it was shown that the concordance between pathologists looking at the same samples wasn't nearly as high as you'd expect it to be. And that has nothing to do with the quality of the pathology, but the complexity of the heterogeneity in these samples. And so the new generation of pathologists said, well, we should start looking at making these measurements. The first thing that had to happen for them to be involved was the FDA had to approve of the first platform for doing diagnoses. And that's been done, and multiple platforms are now improved. And then it became feasible, once you had a digital image of the path sample, to start doing quantitation. And it started out doing very simple thing, counting the number of cells in cell division, counting the number of T cells within a region relative to tumor cells. These are pretty straightforward measurements, and they're also kind of tedious for the pathologist to do themselves, which they had done in the past. Once that was shown to be useful, this first generation, the so-called computational pathology we've been talking about, then started demonstrating capability. And in fact, the FDA has approved the first couple of tests based on computational pathology. And so that's made a major move forward. And then I would say this next generation that we've introduced of, so we're calling it precision pathology because we've taken it a step further in making it unbiased and explainable.
Yeah, I think I said this to Dusty previously that I was speaking to somebody that just came out of school on the radiology side, and she was essentially telling me, "Look, you can't take away, what we have in terms of digitization and in AI and radiology. It's something that we feel like we need to have now." And because there's this general discussion, which I think you guys hit on earlier, which is, hey, is AI going to come and take our jobs away? But as we're seeing, data just continues to grow at an exponential rate, and you just have to have people that are helping to interpret the results.
Don, I was going to add that COVID, the pandemic, has changed some other practices. So there's a relaxation from FDA as to how and when you can diagnose. So you can test it, you can just on your laptop without having necessarily an approval from FDA. So that also increased the acceptance of checking out and being able to view these images on digital platforms and be able to make diagnosis. So that had impact in the last 2 years.
So important. So Dusty, I'll come to you. So, in terms of in radiology, the fact that images were already digitized and accelerated the adoption of AI, did a similar thing happen in pathology? Struggling
Well,
to
yeah,
get the words out, but
as
I, yeah,
as Lance, just said, right? Pathology kind of followed radiology in terms of digitization. They were a little bit behind, but then I think it was 3 or 4 years ago when FDA actually said that you could, read from digitized images. That's when it really took off, and that's where you see the emergence of a lot of these AI-powered pathology companies, which are currently, as Lance said, mostly first generational in terms of capability. Most of them don't really look at this from an explainable AI perspective, and the spatial element seems to be missing in a lot of cases, and that's where SpIntellx has come in and really taken this to the next level with explainable AI and understanding deeply spatial biology or spatial analytics. Lance, you have anything to add since you have seen the field for a long time?
Yeah, no, I think rather than adding to that, because I think you summarized it well, I just want to pick up on something you mentioned earlier, Dusty. Where is all of this going? I hope I didn't take one of your questions away from you, Don. Uh,
no,
and that, what is the potential outcome from where we are in using machine learning and artificial intelligence? I think the future of precision medicine, and this is just a personal view, other people share it, but is that the so called patient digital twins are going to be emerging. Of course, digital twins as a technology have been around for a long time in various industries, but now there's a growing amount of clinical data on every patient can be managed over time for that patient. And now we also are in the realm where using induced pluripotent stem cells and organoids, you can actually create what I like to call biomimetic twins of the patient by making a patient on a chip. And that technology has begun to explode. And now we have the potential to have the actual patient data, and you can't do experiments on patients, but you now have the biomimetic twin that you can do experiments on, test drugs on, and do other manipulation. Wrapping all around that is machine learning tools to integrate all of this data and explainable AI to bring that patient to kind of a point. Now, this is a moonshot. I would say maybe it's a Venus shot, but it's something that we have the beginnings of the tools to approach. And I think more and more places will begin putting this together. And because of the spatial element of doing pathology, precision pathology will be a central component of this direction.
yeah, I've had Trivia Frazier on the podcast before. She's the CEO of Obatala Sciences. They have organoid on a chip, sort of technology, and for sure, I could see the future of what this might be able to contribute in general to the field, especially if you could have digital twins of patients that are maybe you have multiple routes of care that you could possibly go down. Wouldn't it be nice to be able to test some of them in the future?
Yeah, that's actually coming to life as we speak.
Very, very exciting.
Mhm.
Chakra, anything else that you would add?
I was going to say that clearly there is this multi-dimensionality of this data coming from many different places, and it would be nearly impossible without a computational aid to process all this information and get a holistic sort of understanding of this data set. So that's another place wherein I don't think so you can live without those computational aids. And then the question is that how good are these aids and what exactly are they adding to your decision making? Are they helping you get to the next place, or are they stopping you or slowing you down? And I think the explainability there again makes sense across these, all these domains that we mentioned.
Yeah, and it seems like I mean, in terms of literature, spatial biology is pretty. something that a lot of people are taking a look at as well. I guess, why now, I guess, is the key question that I would have. Chuck?
Yeah,
Chakra, you want to go? Or Lance? Yeah, go ahead.
yeah, that's a great question. It is surprising how something, once people start doing it, how obvious it is, but what the mountain that has to be climbed to get people thinking like that. So in pathology, pathologists knew they had to scan a slide and look for patterns. So it's been clear for a long time that the spatial relationships in standard transmitted light H&E staining is important. What's happened, and maybe what's really triggered this, is the development of these multiplexed and hyperplexed labeling schemes. And as Chakra mentioned, there are now 4 or 5 very good platform companies that can produce multi or hyperplexed labeled samples from just 3 or 4 biomarkers all the way up to 100 biomarkers. So you can really define in molecular terms the patient sample on a slide. So then the challenge is, okay, how do you capture all of that information in an unbiased way, define the spatial relationships, and how do you explain what's actually there and how does it relate to the disease? So probably the development of the imaging platforms with the different reagents was the real trigger that opened the door.
Very good. Chakra, did you have anything to add? I thought I saw you start to answer as well.
No, I think I was going to second that, and again go back to the idea that how in the case of tumor, in the case of cancer, what is the host response, and the host response in the communication, which involves communication between cells, and this communication is spatial. So there's no going around it, and the idea is how well can you capture this communication, and how do you the emerging patterns of this communication, and hence this additional interest in spatial biology now, and knowing how something that Dusty mentioned before, the heterogeneity is killing, and the fact that the drugs don't work uniformly well. So knowing the microenvironment and understanding the microdomains has become the critical aspect, from immunotherapy to all the standard therapies out there, and hence this addition. I feel that there's additional focus now on spatial biology to really understand the microenvironment.
Maybe, Chakra, we can get a little bit more granular here. So, talk about immunotherapy and the failures in immunotherapy right now, and why you think understanding the spatial environment, understanding a little bit more from an explainable standpoint, the functionality of cells, if you will, how their development, how is that going to help in today? The response rate for patients with cancer, stage 4 cancer, to immunotherapy is less than 20%. How is that going to help in improving that?
Yeah, I was thinking of an example. We talked about microdomains. So, tissue lymphoid structures, TLS. This is something that people have noticed, renal cell carcinoma and other places, wherein they actually count the number of tissue lymphoid structure, and they found a prognostic value for how many of these tissue lymphoid structures are there. And what they are a collection of the T cells and the B cells, but nobody knows what the role really of these cells are. When are they suppressive? When do they promote cancer? And so this really requires you to understand the spatial nature of the interactions between these cells and with the cancer cells right outside or right surrounding them. And understanding that network biology definitely means that you have found a way to explain the behavior of these microdomains in the form of tissue lymphoid structures, let's say, in predicting the response of this patient to a particular therapy. And this, I think, has become the key across all therapeutic modes.
Yeah, and I mean, so one follow up that I would also have is, what feedback do you have from customers, Chakra?
Yeah, so two things that we noticed with the first generation platforms. One is that if you look at this continuum of imaging, You have the transmitted light platforms that generate transmitted light images, the HND and the IHC. And then you have these other platforms which generate the multiplex and hyperplexed data sets. And it was clear to us that we need solutions that can cover this continuum of data sets. So we have two offerings, HistoMapper and TumorMapper. HistoMapper is for the transmitted light data sets, and TumorMapper is for the multiplex and hyperplex data sets. And what the customers have wanted up until now, and I think that will be the trend, is that they want to be able to use both the platforms in understanding the tissue, because each one of these platforms is giving you different information, morphological ideas with the transmitted light, and the biomarker expression, what is the spatial statistical relationship between the biomarkers. And on the transmitted light side, it could be what are the histological structures and what sort of modeling can you do of these histological structures and the progression with respect to the progression of the disease. So the customer feedback really is that use of both these platforms in analyzing and extracting knowledge of the data sets.
And maybe Lance, you can talk a little bit about the colorectal cancer results that you have, where you actually had a direct comparison between what you guys do and the results from genomics.
So sure, one of the projects that we did was on colorectal cancer, and one of the challenges was in being able to make decisions on recurrence. And using the primary tumor, we were able to look at in retrospective study, which was the first one that was done, we could identify those characteristics of these microdomains and the biomarkers and their interactions that were consistent with of recurrence within 8 years, and those that were consistent with no recurrence over that period of time. And digging deeper within these microdomains using what we call systems pathology, we can define the network biology within these microdomains and actually identify key changes that occur that are consistent with recurrence or not. This then changes the approach that you would take in therapeutic strategy. And actually, one of our collaborators wanted to be able to have a test to choose what treatment would go to what patient based on what is predicted. Because up until that time, it was basically hit or miss. And you know that from, it sounds like your own experience in your family with cancer. A lot of time, the patients are the experimental objects. Because they don't know what the optimal treatment would be. If treatment A doesn't work, then you go to treatment B. So a great opportunity here is to be able to make predictions about what's going to happen that can then guide the therapeutic strategy for those individual patients. And in fact, in terms of the how well the precision pathology worked compared to genomics test, the presently reimbursed genomics test had a so-called area under the curve, which is kind of one of the metrics used, of about 0.72, which is good enough that it was approved and reimbursed. People use the test, but using the SpIntellx approach of unbiased spatial analytics and explainable AI, we could push it over 0.9. And so I think the impact is significant, and that even brings bigger focus to the fact that spatial relationships are important. And just sampling the different regions of genomics, but at what I would call low spatial resolution, is not powerful enough, which is why people started to move to single cell genomics. But you'd want to do that relative to a high resolution spatial biology approach. And that's what we bring to the table.
I think the follow-on question that I would have for you, though, too, Lance, is where do you see this going next? I mean, what then happens, and how does this evolve going further?
Well, I think this is going to be one of the important tools in precision medicine, where a patient is going to be analyzed, and treatments designed in advance based on their data. And I think is cancer as an example, we'll stop doing the experimental testing and try to identify those patients based on their genetic makeup and environmental characteristics have a higher probability of responding to drug A than another subpopulation. So it's going to improve patient outcomes as well as quality of life in cancer going through whatever treatment there are. The other thing is kind of using the same technology, one can select optimal cohorts for clinical trials. So we know that a lot of these drugs that don't get approved did have an effect on 20, 30, 40% of the population of patients, but they were random population. If you could identify cohorts of patients that have characteristics that increase the probability that the drug will have an effect and the patient will respond, you'll collect those patients and make a clinical trial cohort. Now we can have drugs that are really targeted to the right subpopulation of patients. And as Chakra already said, going along with that, the same technology can be used to develop a companion diagnostic, which, especially in cancer, this is something the FDA is very interested in. But it's all from that unbiased spatial analytics and the explainable AI.
So I want to start wrapping us up, but in terms of just other follow-ons, Chakra or Dusty, do you have anything to add to that? I do have one last question that I want to come to, but anything else to add on this one?
Well, I think in general, what I would say is that, Don, we have talked about this. This is the century of biology, right? When Sam Hanash and I were at the last podcast, we talked about the fact how the march of discovery is continuing through the century, right? And if you recall, 2003, when the Human Genome Atlas was published, the headlines in New York Times and Time Magazine was that we have unraveled the mysteries of life. The picture of Francis Collins and Greg Venter,
Yeah,
right?
right.
So, and we know now that we barely scratched the surface with genomics, right? So now, as
Who
we are
would have thought that
progressing,
humans are more complex, Dusty?
humans are way more, and cancer is a very complex disease, right? So,
Right.
as we are looking into genomics from a point of view of mutation, methylation, RNA expression, proteomics, looking at metabolomics, looking at the microbiome environment around the tumor, the microenvironment of the tumor, and looking at pathology in terms of development of the functionality of cells in the tumor microenvironment, combining that with robust explainable AI and powered by next generation computational technology, whether it's quantum computers, you might need very powerful computers to do all the computation ultimately when you have all this information together. That is going to result in the outcomes that Lance is talking about. And then, of course, once you develop the digital twin based on all this information, you can actually have interventions on the digital twin to see the efficacy or the safety of drugs. So that's the promised land that we are working towards. Hopefully in the next 10 years we'll get closer to that.
Yeah, it's well, I mean, I was talking to somebody earlier today about Amazon's compute, because they constantly, whenever you need more compute power, you just turn to Amazon and just say, hey, I need more compute, and they turn it on. So this idea of having supercomputers and other things, becomes much more of a reality for people that are developing fabulous technologies. And so, Chakra, I would come back to you maybe for my last question here. Um, as the CEO of SpIntellx, how do you see this evolves with regards to the patient, and how things get used into the future? Where do you see the clinical workflow go for patient outcomes?
Yeah, happy to respond to that, Don. So, explainability, we focus a lot on that and how it's going to change the workflow. One way to put all this in context is if you have a cohort whose clinical profile is all roughly the same, their molecular phenotypes are roughly the same, some responded to therapy and some did not. there's nothing you can do. And that's when I think by looking at the tissue, by doing this unbiased spatial analytics and explainable AI, we have this power uh, to dig deeper into why some responded to therapy and some did not. And I think this basic idea will pervade all the application scenarios that we painted, whether it is companion diagnostics or whether it is drug discovery or suggesting novel combination of drug. This idea that you look at space and you have to look at the space with the idea that biology is going to emerge and you're going to capture this emergent biology in an unbiased manner. And I think that's where this is going to head up.
And on just in breast cancer, there are 700,000 biopsies done every year in the US. And if you look at all cancers, probably a few million. And I would say that at this point, we use less than 1% of the information that is extracted from them in standard pathology and also by the Gen 1 AI and AI pathology tools that we have. So we got to go beyond that.
And so that's definitely where this could contribute. Lance, how about from you?
So this is even going to permeate the practice of pathology, whether it's in medical centers or in private practices. We have a collaboration with a large West Coast pathology practice with greater than 60 pathologists. And one of the challenges is how do they get, and they have specialists, subspecialists, as well as generalists, how do they get the right patient sample to the right pathologist? And with a workflow in place where the images are managed, the metadata is managed, the digital images are managed, and with the application of the precision pathology, initial decisions are made and they can be directed to the right pathologist who can then look at the data, ask the question why, make sure they agree with what the algorithm has said or not. The efficiency is going to go up dramatically. Now, the pathologists aren't going to be replaced by these tools, but their talents are going to be focused on the more difficult, challenging samples. And so the efficiency and accuracy will go up. We've already done some studies on some early work that showed both of those occurred. And the patients will be happier because they'll get a faster, more accurate diagnosis.
Yeah, so thank you so much. Any final comments from anybody before I wrap us up?
Well, this was a great conversation. I'm glad that I got to be in this with the folks from SpIntellx, and really appreciate Don giving us this opportunity.
Thanks so much. And so, Lance, Chakra, and Dusty, thank you so much for being a part of the Life Science Success Podcast. I greatly appreciate you being here.
Don, we enjoyed it. Thank you.